All right, everybody. So one of the ways that the endocrine system uses your ATP is through this purinergic signaling, and purinergic receptors interact with extracellular ATP. And in your posterior pituitary gland, you have these P2X receptors, which are your some purinergic receptors. They're trimeric membrane-bound ion channels. And so, in regards to vasopressin, which is one of the hormones released by the posterior pituitary gland, what will happen is that ATP will come in and bind extracellularly to these P2X receptors, which causes the channels to open. And so calcium will then rush into the cell and cause your vasopressin containing vesicles to begin to move towards the membrane so that vasopressin can be released into the bloodstream. And vasopressin helps regulate water retention in the body. So the more vasopressin you have in your bloodstream, the greater water retention you'll have, which will increase your blood pressure. And we've also got intracellular signaling. So you get your ATP from glycolysis, the citric acid cycle, oxidative phosphorylation. And so this ATP in your pancreatic beta cells will inhibit these ATP sensitive potassium channels, which causes them to close. And then membrane depolarization occurs, which causes these calcium channels to open. And again, calcium will rush into the cell and cause these insulin containing vesicles to begin to move towards the membrane so that insulin can be released into the bloodstream via exocytosis and regulate your blood sugar levels.
So the next use for ATP that we found in the endocrine system is the calcium regulation of intracellular calcium levels, as well as secretory vesicles. Starting with the secretory vesicles, after a vesicle is secreted from the Golgi apparatus, as you can see in the figure, it is transported down a microtubule towards the cell membrane. And this is done by a motor protein, specifically a kinesin, which uses ATP hydrolysis in order to power its conformational changes slash walking movement. Following transportation by the kinesin protein, the V-snare and T-snare proteins, which are activated by intracellular calcium levels, attach the vesicle to the membrane. The activation of V-snare and T-snare proteins are caused by the interaction between synaptotagmin and the intracellular calcium. When that calcium binds to the synaptotagmin, it helps to bind the V-snare to the T-snare protein, which pulls the vesicle in closer to the membrane so the two membranes can begin to fuse together. After those membranes fuse together, the hormones can then be released. Following exocytosis, an ATP-powered calcium ATPase restores the intracellular calcium levels back to normal by rapidly pumping the calcium out of the cell.
Okay, we're going to talk a little bit about ATP and its role in vitro fertilization. So sperm have to go through a lot of reactions really quickly to fertilize an egg, and one of them is called the acrosome reaction. And then the acrosome reaction is actually where the head of the sperm is changed in a way that the sperm can physically fuse with the egg. And that's kind of what allows for fertilization to occur. And you can kind of see in that diagram there how the sperm will actually penetrate through the outer membrane of the egg and into the inner layer of the egg. And so during IVF, we have to kind of mimic the conditions inside of the body to make sure that the acrosome reaction can occur properly, as well as the other reactions that have to occur. And so one study found that the ATP, when the sperm were placed in a solution containing high amounts of ATP, that sperm had the highest rate of the acrosome reaction compared to their other solutions. And so this demonstrates that ATP can be used in combination with IVF to achieve the most successful results and the best rates of fertilization inside of the target cells.